Hello everyone. Our group will focus on the study of Ngogo et al. 2015, which enhanced bioremediation of soil artificially contaminated with petroleum hydrocarbons after enactment with Capra, aegagrus, hircus, manure. Pollution with crude oil and its derivatives have become a major environmental concern as they impose a huge threat to the natural ecosystems and human health. This is due to their potential toxicity and carcinogenicity when present in high concentration. Therefore, efficient remediation of crude oil contaminated soil is needed for the normal functioning of biological and ecological systems. Bioremediation is regarded as the most desirable procedure for remediation of oil-contaminated soil because it is efficient, inexpensive, and applies environmental-friendly processes compared with the physical and chemical remediation approach. The most frequently used technique in bioremediation involves the enhancement of the activities of the microorganism through stimulation with nutrients, which is known as biostimulation. Nitrogen-rich organic substrates such as animal manure, coconut shell, and rice husks have been shown to increase the rate of bioremediation when used as biostimulants. Therefore, this research work aims to explore the effectiveness of the goat manure toward enhancing the bioremediation of the crude oil contaminated soil. The amended sample and control sample were analyzed for several physical-chemical parameters at 14 days interval during the study period. Furthermore, the total petroleum hydrocarbon concentration in both soil samples were quantified through gas chromatography. The total culturable heterotrophic bacteria and culturable hydrocarbon utilizing bacteria were isolated and identified based on their colonial and microscopic morphology and biochemical characteristics with reference to the virgin manual of determinative bacteriology. Lastly, for the TPH reduction, the degree of the significance was determined using the one-way analysis of variance.
Analysis of physical-chemical parameters at 14-day intervals show that pH of the Ca hircus manure, pristine soil amended samples and unamended samples were all slightly acidic. However, in the first 28 days, pH of the amended and unamended samples slightly increased to alkaline before gradual decrease was obtained in the end of study period. It was obtained the total organic carbon of amended sample was decreased by 16.41% and unamended sample was decreased by 4.6%. Similarly, there were decrease in concentration of nitrogen, phosphorus, and potassium in both amended and unamended samples, in which the percentage of decrease in amended sample is comparatively higher than unamended sample. Next, there were significant differences in TPH concentration at different time intervals in the amended sample. In general, the decrease in TPH concentration of amended and unamended samples were 62.08 and 8.15% respectively by the end of the study. Also, microbiological analysis of amended and unamended samples show steadily increase in colony-forming unit for both total culturable heterotrophic bacteria TCHB and culturable hydrocarbon utilizing bacteria CHUB. This study also identifies some genera from bacteria isolates, includes Acinetobacter, Achromobacter, Bacillus, Flavobacterium, Klebsiella micrococcus, Pseudomonas, and Staphylococcus. Among the isolates, Micrococcus and Pseudomonas were predominant bacteria isolates, while Achromobacter and Flavobacterium were less frequent isolates.
In conclusion, this study demonstrated that the manure is a good organic substrate containing nitrogen, phosphorus, and potassium which have great potentials for enhanced bioremediation of crude oil contaminated soil. The manure is a good biostimulant which enhance the activities of indigenous hydrocarbonoclastic bacteria, resulting in significant decrease in TPH concentration of crude oil contaminated soil. The use of the manure can be a cost-effective and process-efficient bioremediation option for hydrocarbon-contaminated soils.
However, this study has its strengths and limitations. For the strengths, utilizing the GOTS manure is cost efficient and process efficient. For example, fertilizer has been conventionally used as a biostimulant of petroleum hydrocarbon polluted sites. However, it is more expensive and it shows lower petroleum hydrocarbon reduction, and it brings negative consequences to the environment. Therefore, GOTS manure has the advantage of being readily available, and it only utilizes the naturally occurring microbes in soil. Secondly, the Capra aegagrus hircus manure able to provide nutrients. The growth and activities of hydrocarbonoclastic bacteria requires phosphate, carbon, and nitrogen to degrade the contaminants, whereby the GOTS manure able to supply all the building blocks needed for that. Thirdly, the experimental design increases the bioavailability of microbes. Experiment design involves treating soil samples added with crude oil, rather than using the contaminated aged oil. And this shows increases bioavailability of hydrocarbons to microbes, where the crude oil is a surfactant that increases the mobility and rate of biodegradation.
Next, moving on to the limitations of this study. Firstly, using manure as the biostimulants might show different results. Why? This happens if the god has a deficiency of nutrients in his diet, then it will cause the manure to have low nutritional value, where it would affect the biostimulation results eventually. Besides, this study does not clearly state the method to mix the excavated soil sample. Therefore, if the researcher only mixes the composite sample in the bucket, then we suggested the researcher sieve the soil samples on a 2 mm sieve to enhance proper mixing and extract consisting mainly of stones and dead plant debris discarded. Moreover, this study is easily affected by abiotic factors such as temperature, soil pH, and humidity that control its potentiality, where these factors are difficult and unable to be controlled. To improve this study, we suggest the researcher use naturally contaminated soil. This is because the naturally contaminated soil is bioremediated more as compared to artificially contaminated soil. In addition, this study would benefit from being conducted with a research design of 3 replicates, as 3 replicates of an experiment is generally considered the minimum. Overall, this is considered a thorough paper, and the writing style of this research paper is clear and well organized. However, some improvements still can be made to the method to permit greater results. These are the references that we use for this presentation, and that's all from us. Thank you.